expression. Nilagay ko yung League of Legends dyan kasi habang naglalaro tayo, di ba, ng computer games, meron tayong controls. Kawat daa, sabi ni Jeffrey. Tama, kawat ini. Okay. Meron tayong controls, di ba? Merong 4 na abilities yung chosen character mo. So, for example, pipinutin mo yung R, yun yung pinakamalakas niyang ability. Nakakontrol mo kung ano yung abilities ng characters mo para labanan mo yung kabilang kampo. So, in this way, parang meron kang control on which abilities you express. And ngayon, baka sinisira ko ng League of Legends sa inyo. Maalala niyo na yung League of Legends. Maalala niyo na yung control of gene expression habang naglalaro kayo ng LOL. Kasi, again, ito, pag-express siya ng abilities. So, today, we are going to take a look at bacterial gene regulation and eukaryotic gene regulation. Isahin natin tong dalawa. And first, we must define some terms. So, gene regulation, in gene regulation, we have these kinds of terms. Or in genetics, molecular genetics, we have these terms, constitutive genes, which codes for proteins that are always active in growing cells. So constitutive genes, they are the genes which are always active in the growing cells. So ano kaya yung mga proteins na to na parating active sa growing cells? I-assume natin na ginagamit sila for translation ng proteins or transcription kasi parati tayong nagta-translate ng proteins sa ating cells. So, ribosomal genes are an example of constitutive genes. Kasi again, parati natin kailangan ng proteins. Therefore, parati natin kailangan mag-translate ng proteins and ngayon, kailangan natin ng ribosomes. And yung ribosomes na yan ay ina-express ng ribosomal genes. Yung pangalawa ay tinatawag na regulated genes. These regulated genes encode enzymes for metabolic processes that are not constantly required. So, kaya sila tinatawag na regulated kasi hindi naman sila kailangan i-express parate. And meron tong parang way para magkaroon tayo ng tinatawag na adaptive enzyme synthesis. Kasi itong regulated genes, depende ito sa environment ng cell natin. So, kung merong sa environment ng cell natin na kailangan na pwedeng magamit para mag-produce ng certain kinds of proteins, saka lang ito ma-activate na regulated genes. Maintindihan natin to maya-maya. So, proceed muna tayo. So, review lang nung dalawang pathways in metabolism. Alam niyo na to, catabolic or degradative pathway. Yung nangyayari, nasi-separate mo yung isang malaking molecule into monomers of that molecule. And yung isa ay anabolic or synthetic pathway. And in anabolic or synthetic pathway, sabi ng mama ko, mabilis daw ako magsalita. Mabilis ba ako magsalita? Wait lang. Sige, bagalan ko tuloy. In anabolic or synthetic pathway, nagsisynthesize tayo ng bagong molecules. So, difference again ng catabolic sa anabolic pathway. Sa catabolic, nagde-degrade ka ng molecule. Sa anabolic, gumagawa ka ng molecule. Alam niyo na yan. So, unahin natin yung catabolic pathways. Catabolic pathways, they are related to substrate induction. So, ano ba tong substrate induction na to? First, let's define some terms. Catabolic enzymes are enzymes which degrade specific substances to obtain energy. Yung mga catabolic pathways natin, most commonly, ginagamit sila kapag kailangan natin ng energy. Kailangan natin mag-breakdown ng energy. For example, di ba, pag kakain kayo, di ba, yung rice na kinain nyo, mabi-breakdown siya sa katawan nyo. Yung mga sugar sa rice nyo, bine-breakdown siya. Catabolic pathway yung ginagamit para makuha nyo yung energy from your rice para magamit ng body. So, yun yung catabolic pathway. And yung mga enzymes involved doon ay yung catabolic enzymes. Substrate induction naman, meaning turning on of enzyme. So, when you say induction, yung meaning lang nun ay to turn on that enzyme. And you will see later that in substrate induction, you use a certain type of substrate to turn on the enzyme. And di ba, alalahanin nyo, kapag merong substrate, merong enzyme. And yung substrate na naka-pair doon sa enzyme na yon, siya rin yung nagto-turn on ng gene para doon sa substrate na yon. Wait lang. Inducible enzymes naman are enzymes whose synthesis is regulated in this way. So, inducible enzymes, meaning lang nun, yung mga enzymes na nai-induce, na to turn on sa pamamagitan nito. So, ayun. Catabolic pathways and substrate induction, yung example natin na medyo familiar na kayo, ay yung lactose metabolism. Wait lang. Ay yung lactose metabolism. So, in lactose metabolism, what you need to remember is that, sa bacterial cell to ha, what you need to remember is that the lactose needs this protein named galactoside permease para makapasok yung lactose from outside to inside of the cell. Para maalala nyo yung galactoside permease, nasa pangalan niya na mismo, permease. Ibig sabihin, nag-permit ng entry ng lactose. So, galactoside permease, remember this protein, it allows the lactose to move from outside to inside of the cell. And once na nasa loob na ng cell yung lactose mo, yung enzyme na nag-act upon your lactose mo ngayon ay yung tinatawag mong substrate, and yung enzyme na nag-act upon lactose is your beta-galactosidase. And beta-galactosidase, yung mas madaling term lang actually sa kanya, lactase. Pero beta-galactosidase para mas social. Ayun. Beta-galactosidase is the enzyme responsible for breaking down lactose into galactose and glucose. Okay. So, tandaan nyo itong dalawa ha, galactoside permease and beta-galactosidase. Tingnan naman natin yung anabolic pathways and end product repression. In anabolic pathways, yung ginagawa, di ba, synthesis. So, you make a product. End product repression, meaning you use the product itself to turn off the production of the product. So, dito naman, tiniturn off ng product mismo yung paggawa ng sarili niya. At nangyayari yon kapag malaki na yung amount ng product sa loob ng cell, as hindi niya na kailangan mag-produce ng product na yon. Kaya kailangan niya na i-turn off yung paggawa ng product. Repression is a general term in molecular genetics referring to the reduction and expression of any regulated gene. So repression, general term siya para lang sabihin na turn off mo yung gene na yon. And this has an effect on protein synthesis and protein activity. So dinidefine pa lang natin yung mga terms para mas madali na mamaya. The one thing that is common in both catabolic and anabolic pathways are the effectors or effector molecules. Both anabolic and catabolic pathways, they have effector molecules, which are small organic molecules that trigger induction or repression of enzyme synthesis. Induction or repression. So, for example, yung effector molecule mo dito ay itong square things, square small things. Tapos, magbabind siya ngayon dito sa enzyme or in protein. Tapos, ito ngayon ay i-induce or mag-repress ng enzyme na yon. So, again, yung effector molecules, common siya in both catabolic induction and anabolic repression of the gene of an enzyme. Ayun. And gumawa ko ng table para mas madali nyo maalala. Kapag yung pathway ay catabolic, yung effector molecules niya ay yung substrates niya. And yung function ng effector ay para mag-induce ng production ng enzyme. Turning on. Pero kapag yung pathway ay anabolic, yung effector molecules niya ay end products, and yung function ay mag-repress ng production ng enzyme na yun, nagagawa ng end product. Mas maalala nyo ito mamaya pag nakita nyo na yung example natin. So, una, tingnan mo natin yung history ng lahat ng to. So, kailan kaya nagsimula itong aralin itong bacterial gene regulation? 1961 lang. So, imagine niyo mga 60 years ago. 60 years ago. Si François Jacob and Jacques Monod, they are French scientists. They actually studied the operons in your E. coli. So they studied operons in E. coli, and these operons, they found out that the genes involved in lactose catabolism are organized into an inducible operon. So first, let's define an operon. So narinig niyo na ba yung word na operon? Pero ayun, yung operon, ibig sabihin lang yan, collection siya ng related genes. So sa isang mahabang stretch, merong related genes doon na magkakasama. Ang tawag doon operon. And sabi ni Jacques Monod and François Jacob, sabi nila yung genes daw for lactose catabolism in bacteria are organized into an inducible operon. And remember, yung inducible, yung meaning niyan ay not pwede ma-turn on. So, let's take a look. So, ito yung diagram ito yung itsura ng operon, ng lac operon sa eukaryotes. Sa eukaryotes ulit. Sa E. coli, sorry. Ito yung itsura ng lac operon sa E. coli. So, merong promoter region. Sa promoter region, daan nagbabind yung polymerase. Sa operator, dyan nagbabind yung repressor. And meron kang yung LAC-Z gene, yung LAC-Y gene, and yung LAC-A gene. And itong kabuuan na to, ito yung tinatawag na lac operon. Again, operon kasi itong genes na to, itong LAC-Z gene, LAC-Y gene, and LAC-A gene, they are related. How? They are related in lactose catabolism. You will see later on that the LAC-Z gene produces the beta-galactosidase. And beta-galactosidase, remember, yung function niya ay to catabolize yung lactose and separate them into yung monomers ng galactose and glucose. And yung LAC-Y gene naman ay nagpo-produce or ang ina-express niya ay yung galactoside permease. And galactoside permease is again needed for the lactose to get from outside of the cell to inside of the cell. And ito lang yung dalawang kailangan yung alalahanin. Huwag yun na ito alalahanin na transacetylase. Yung transacetylase, di naman yan masyado kailangan nyo alalahanin. Function lang yan, maglagay ng acetyl group doon sa lactose. Pero huwag yun na alalahanin. Ito lang yung alalahanin nyo. Beta-galactosidase and galactoside permease. And upstream of the lac operon is a regulatory gene. So nasa taas ng lac operon, meron daw na regulatory gene. And this regulatory gene produces a repressor protein. From the word itself, repressor protein, maisip nyo na yung function niya ay to repress. Ano yung nire-repress niya? Ang nire-repress ng repressor protein ay itong lac operon. So when this repressor protein binds to the operator of the lac operon, it represses the expression of the lac operon. Tingnan natin. The lac operon is negatively regulated now by the lac repressor. So nakita natin, di ba dito, meron repressor. Ito yung repressor. And this repressor, it negatively regulates daw the lac repressor. So how? So, pag natin to tingnan, bakit kailangan natin alamin kung paano kinokontrol yung lac operon. Kasi in bacteria, yung favorite na pagkain ng bacteria ay glucose, di ba? So, alam natin na yung favorite na pagkain ng bacteria ay glucose. Pero, in the case na walang glucose sa environment, walang available na glucose sa environment, therefore wala silang makain, kumukuha sila ng parang mga substitute. And that substitute is your lactose. So, ngayon, itong lactose metabolism, nangyayari lang siya in an event na walang glucose doon sa environment ng bacteria. Kaya natin to inaaral. So, let's proceed. So, sabi daw, yung repressor protein, it negatively regulates the lac operon. And the repressor protein is bound to the operator. Once the repressor is bound to the operator, the RNA polymerase is blocked from transcribing the lac operon. Hindi siya makakagalaw. Hindi makakagalaw yung lac operon. And kailan to nangyayari? Kailan na-repress ang lac operon? Sabi natin, yung favorite food ng bacteria ay glucose, di ba? So therefore, hindi mo kailangan ng lactose metabolism kung merong glucose sa environment ng bacteria. So therefore, nagkakaroon ng repression ng lac operon kapag available naman yung glucose sa environment ng bacteria. Okay? Sana na-gets niyo yung sinasabi ko. Ayun. The lac operon is not transcribed. Again, bakit? Kasi di mo naman kailangan mag-transcribe ng lac operon para magkaroon ng lactose metabolism kasi meron ng glucose sa environment ng bacteria. Hindi niya na kailangan yung lactose. Parang second choice lang tong lactose para lang kapag may time na mawala ng glucose sa environment. Okay. So, lactose, no. Repressor, yes. Transcription, no. Dapat nilagyan ko rin dito, glucose, yes. Merong glucose sa environment. Ito naman, this happens when lactose is available in the environment and wala kang glucose. So, sa event na to, walang glucose sa environment. So, yung second choice ng bacteria, gamitin yung lactose para makakuha ng glucose. Diba kasi yung lactose, kapag nakatabolize siya, nagiging galactose and glucose. And nandun yung glucose, gagamitin ng bacteria, yung glucose. So, yung glucose ngayon, yung substrate, it binds to the repressor protein and changes its conformation. When a protein changes its conformation, ang nangyayari, nababago rin yung function niya. Or hindi rin siya nag-function ng maayos. So therefore, yung repressor protein mo ngayon, since nagbago yung conformation niya, hindi na siya makakabind doon sa operator. And now, your RNA polymerase has the ability to transcribe the lac operon. Again, yung LAC-Z gene, it produces the beta-galactosidase, which catabolizes yung lactose mo into two monomers of galactose and glucose. LAC-Y gene is for the production of galactoside permease, which permits your lactose from entering outside of the cell to inside of the cell. And yung trans-acetylase, huwag yun napansinin. So, ayun. Nangyayari to kapag walang glucose sa environment ng bacteria and merong lactose. So, walang glucose? Wala. May lactose? Yes. May repressor? Wala. Kasi yung lactose, nakabind siya doon sa repressor protein and na-change yung conformation. Merong transcription? Yes. Okay. Another type of regulatory mechanism in the lac operon is through the catabolite activator protein, or CAP, which positively regulates the lac operon. And di ba kanina negative regulation, so merong repression na nangyayari, merong turning off na nangyayari. Dito naman yung mangyayari, turning on of the gene or the lac operon. So sabi dito, meron kang CAP protein or catabolite activator protein or CAMP receptor protein that turns on transcription. So how? It actually couples with cyclic AMP or CAMP which is a secondary signal that reflects the level of glucose in the cell. When CAMP is at high concentration when the amount of glucose is low. Again, kapag yung environment ng bacteria mababa yung amounts ng glucose, walang glucose sa environment ng bacteria, kailangan niya i-transcribe yung lac operon para magkaroon siya ng second source of energy, which is your lactose. So, in the event na mababa yung concentration ng glucose, nagkakaroon ng maraming production ng cyclic AMP. Therefore, yung cyclic AMP, ginagamit siyang parang indication na kung ano yung level ng glucose doon sa cell. And this cyclic AMP binds to the CAP protein and forms a complex. And this complex can bind to an area upstream of the promoter region called your CAP site. And sa CAP site, nagbabind yung complex. And the CAP complex binds to the CAP recognition site, seen near the promoter of the lac operon, and this actually promotes translation or transcription, of your lac operon. The promoter becomes more readily bound to the RNA polymerase. So your RNA polymerase now can transcribe your lac operon, and again, it will produce the enzymes and proteins needed for lactose metabolism. The CAP complex, itong CAP complex, sabihin na natin na complex, parang ine-enhance niya yung paggawa ng enzymes for your lactose metabolism. So, ine-enhance nitong CAP complex yung gawa ng enzymes for lactose metabolism kasi nga, mababa yung glucose sa cell. Kaya kailangan niya ng bagong source ng energy, which is your lactose. So, glucose, wala, mababa. Merong CAMP, mataas, yes. Merong lactose, yes. Merong transcription, meron. May video, wait lang. Para mas maintindihan nyo yung CAP, yung lac operon, yung CAMP, ito yung video na kailangan natin paroorin.
Mamula pong audio.
Hi, in this clip we're going to demonstrate the gene regulation of the E. coli lac operon. As you remember from the lecture clips, the lac operon is a collection of genes that produces the lactose metabolism machinery when expressed. Now, when these are expressed, E. coli is able to metabolize lactose. However, it prefers to metabolize glucose to use that as its sugar. Thus, when there's glucose available to the cell, it's pointless for it to create this machinery because it's not going to actually use it. It's going to metabolize the glucose instead. But when we remove glucose and add lactose, the E. coli absolutely needs to be able to metabolize this lactose. Otherwise, it's not going to be able to grow with the available energy source. Thus, we have a couple of different situations. When we have glucose present, but not lactose, we do not want to express the lac operon. However, when we remove glucose and we add lactose, we do want to express the lac operon. Now, we've covered when the E. coli would like to express this operon. However, how can it express this operon only when it wants to and not when it doesn't? That's where the promoter comes into play. The promoter is where RNA polymerase binds, and RNA polymerase needs to bind to this promoter in order to transcribe and express these genes. In this particular promoter, we have a site where a repressor can bind and a site where an activator can bind. So I have a repressor and I have an activator. So when the repressor is bound, polymerase cannot bind to the promoter. Polymerase also needs the activator to be bound to the promoter in order to bind. So how can the E. coli regulate when the repressor and activator are bound to the promoter? Well, when we have lactose in the system, I have lactose right here. The repressor is able to bind lactose. The repressor only binds to the promoter when there's no lactose, and when binding to lactose, the repressor comes undone from the promoter. How does the cell tell whether there's glucose in the cell or not? When glucose is low in the cell makes a molecule called cyclic AMP, or CAMP. I have a molecule of CAMP right here. And so CAMP is the cell's way of indicating that there is low glucose in the cell. The activator needs CAMP to bind to the activator binding site. When CAMP is removed, or glucose is added, the activator cannot bind to the promoter anymore. Okay, so let's give it a try. In this first situation, we have glucose, we don't have lactose. So I have neither my lactose nor my CAMP in the system. Repressor, can you bind? The repressor is bound. Activator, can you bind? No, the activator is not bound. The repressor is bound. And so when RNA polymerase comes to the promoter, RNA polymerase is not able to bind at this promoter. And thus, it's not able to express these genes. Okay, and so, as we can see here, there is no expression. In this second situation, we have no glucose, and we do have lactose. So I'm going to, since I'm removing glucose, I am adding back CAMP. Activator, can you bind? Activator is now bound. And I'm also adding lactose to the system. Repressor can't bind anymore. So what do I have at my promoter now? RNA polymerase comes along and finds only an activator at the promoter. The polymerase can thus bind to the promoter and move downstream to transcribe and express the lactose metabolism machinery that we have here. So we've now covered how the E. coli organism is able to regulate the transcription of the lactose metabolism machinery. This is only one example of many genes in many organisms that are regulated by mechanisms such as this. Gene regulation is a very widespread mechanism in order to keep a cell from producing proteins that are a waste to produce. Thank you for watching.
Yay! So, ayun, di ba? Nakita nyo kung paano sila umacting para para mag-regulate ng lactoferrin. Actually, kung face-to-face classes to, papagawin ko sa inyo yun sa board. Maghahanap ako ng actors, tapos yun. Re-recruit ko sana kayo, tapos mag-regulate tayo ng lactoferrin. Pero since nga, sayang, hindi face-to-face classes, yun na lang muna. Pwede nyo gawin to sa bahay, i-recruit nyo yung kapatid nyo, yung mama nyo, kung sino man. Tapos gawin nyo yun, di ba? Para mas maalala nyo yung gene regulation in the lac operon. So, proceed muna tayo sa ating lecture. Meron ba kayong questions so far? Naintindihan niyo ba or medyo magulo? Kung may questions kayo, pwede kayo magtanong para masagot natin agad. Okay. So, kung wala naman tanong so far, proceed muna tayo. Dito naman tayo sa tryptophan synthesis. And tryptophan synthesis is an example of an anabolic pathway. So, tryptophan is an amino acid, di ba? And if the bacterial cells needs tryptophan, it needs to synthesize tryptophan. And it has been found out that the genes involved in tryptophan synthesis are organized into a repressible operon. So this time, repressible operon daw yung tryptophan. Kanina, di ba, inducible yung lactose, yung lac operon. Ngayon, repressible operon yung tryptophan daw. So let's take a look muna at how kung anong itsura ng tryptophan operon. Again, meron ka ditong promoter region. Diyan nagbabind yung RNA polymerase. Sa operator, diyan nagbabind yung repressor protein. At ito yung genes mo involved in your tryptophan synthesis. So, hindi nyo naman kailangan isa-isahin kung ano yung pinaproduce nitong genes na to. Ang isipin nyo lang, ang genes na yan ay related and related sila sa pag-synthesize ng tryptophan. Kaya sila tinawag na operon kasi these are related genes for the production of tryptophan. And then you have a regulatory gene upstream of your operon, which produces your repressor. And yung tryptophan operon mo, again, yung ginagawa niya ay tryptophan synthesis. Yun lang yung kailangan nyo lalahanin. So when the repressor produces a repressor molecule or a repressor protein and there are available tryptophan in the cell, magbabind yung tryptophan doon sa repressor protein. And that repressor protein will bind to your operator and prevents transcription, prevents the RNA polymerase from transcribing the TRP operon. So in this case, marami kang tryptophan sa cells ng bacteria. So hindi mo naman kailangan mag-synthesize, di ba? Hindi mo kailangan mag-synthesize kung meron ka ng tryptophan. And may repressor, meron. Kasi hindi mo kailangan yung gene for synthesizing tryptophan again kasi meron ka ng tryptophan sa loob ng cell. Merong transcription, wala. Next naman, if walang tryptophan sa loob ng bacterial cells, meron kang inactive form ng repressor and yung RNA polymerase can transcribe your TRP operon. So in this case, kailangan mo ng tryptophan doon sa bacterial cells. So since wala kang tryptophan na available, wala ring magbabind dito sa repressor protein. Therefore, inactive form yung repressor and hindi siya makakabind sa operator. So, ngayon, yung RNA polymerase mo, makakatranscribe siya ng DRP operon into tryptophan. Ayun lang. So, may tryptophan sa cell? Wala. May repressor? Wala. Kasi kailangan mo mag-produce ng tryptophan and may transcription? Meron. So, ayun lang yung mechanisms in bacterial gene regulation. Kailangan nilang parang tandaan ay kung ano yung mga conditions within the bacterial or within the cell. So, kunyari, yung situation, mababa yung glucose, mataas yung cyclic AMP, meron bang transcription, ganon. Malay nyo, lumabas sa exam. Malay nyo lang naman, di ba? So, next na, proceed na muna tayo. Eukaryotic gene regulation na tayo. So, sa eukaryotic gene regulation, medyo mas complex to kasi complex organisms din ang eukaryotes. So, as you can see here sa picture, di ba, maraming organs. Maraming organs. Merong eye, merong lungs, merong brain, merong muka, merong liver, etc., etc. So, sa eukaryotic gene regulation, mas complex. Bakit? Dahil iba-iba yung cell types natin. At iba-iba rin yung pinoproduce. Since iba-iba yung cell types natin, iba-iba rin in-expect mo yung genes na ginagamit sa different cell types. And also, in eukaryotes, remember, yung packaging ng DNA ng genome natin ay iba, di ba? Nakapulupot siya into chromatin or nakapulupot siya into chromosomes. Unlike sa bacteria, mas madali lang kasi bilog lang yung genome nila. Sa eukaryotes, iba dahil iba yung packaging. So, ang dami mo pang challenges kung paano mo ma-express yung genes doon. And ito yung main levels of eukaryotic gene regulation. First is yung genomic control. Second is yung transcriptional control. And lastly, yung post-transcriptional control. So, yan yung tatlong levels, main levels of eukaryotic gene regulation. And let's take a look at each of this. So, di ba nakita niyo may 5? Bakit may 5? Sabi mo, ma'am, 3 lang. Bakit biglang may 5 dito? Makita niyo mamaya. So, una, meron tayong in the level of genome. So, in the level of genome, meron tayong gene amplification, deletion. Huwag niyo to isulat muna. Kung nagdo-notes man kayo, di niyo to kailangan tandaan. May mention ko lang yung parang overview ng gene expression. So, merong gene amplification or deletion sa genome level to. So, therefore, doon sa DNA mismo, yung changes, merong DNA rearrangements, DNA methylation, chromatin decondensation and condensation, histone modifications through methylation and acetylation and changes in the HMG proteins. And yung next level ay yung transcription. And sa transcription, yung main mechanism ay controlled by transcription factors. Yung third level ay yung RNA processing and nuclear transport. And dito, di ba, after mo mag-transcription, gumagawa ka ng pre-mRNA. And yung pre-mRNA, it undergoes RNA splicing and other processing events. Namely, yung pagdagdag ng 5' methyl cap at polyadenylation, di ba? So, yung RNA splicing and pagdagdag ng mga caps na yon, kasama na yon sa RNA processing or sa RNA processing. And also, kasama na rin doon yung nuclear transport kasi kapag hindi maganda yung pagkalagay mo ng 5' methyl cap or wala yung 5' methyl cap, di ba? Hindi matatransfer yung mRNA from the nucleus to the cytoplasm. And so, crucial yung steps na yun. And yung next step ay in the translation regulation. So, in the translation, meron ka ng mature mRNA sa cytosol. Therefore, pwede na siya ma-translate into proteins, into polypeptides. And dito, sa translation control, yung nangyayari, nagkakaroon ka nung mga pagpunta nung polypeptide sa ER. Meron rin ng pag-initiate ng control ng translation, nung initiation factors ng control ng translation, and meron ding mga translation repressors tayong nakikita. And yung favorite kong part actually dito ay yung microRNAs. So, merong function sa translational repression. Yung ginagamit dito ay yung RNA interference na makikita natin mamaya. And lastly, yung isa pang level, fifth level is in the post-translation, wherein yung dinideal with mo na yung protein. So, yung protein mo, pwede mo siya makontrol in folding and assembly, kung paano mo i-fold yung protein, pwede mo through cleavage, pwede ma-modify mo yung protein, and pwede mo rin makontrol kung saan mo i-import na organelles yung proteins. Kasi di ba yung proteins, sila yung functional, may joke na pumasok sa isip ko. Sorry. Sila yung functional na ginagamit sa loob ng cell. And sila yung nagpapatakbo ng halos lahat ng cellular functions. So, kailangan mo sila i-import into their different stations sa organelles para mag-function sila ng maayos. So, bakit sabi ko kanina may 3 levels, diba? Kasi, ito, genomic control. Itong genomic control, dito lang siya sa level of the DNA. Sa level lang ng genome. So, yung changes lang sa DNA, yun yung general nasakop ng genomic control. Yung second, yung transcriptional control. Transcription, meaning yung transcribe ng RNA ay ng DNA into RNA. So dito sa transcriptional control, dito lang siya sa transcription mismo. And post-transcriptional control, kasama tong tatlo. Kasama yung RNA processing and nuclear export, kasama yung translation, and kasama yung post-translation. So sa post-transcriptional control, post-transcriptional, pagkatapos ng transcription, siya, yung sakop niya. So, ayun lang. So, unahin natin yung genome, in the level of the genome. In genomic control, parang history muna tayo. So, noong 1997, 24 years ago, noong 1997, si Ian Wilmot, 24 years? 23 years? Wala na. Si Ian Wilmot sa Roslin Institute sa Edinburgh, Scotland, nang silang ginawang experiment. Nag-clone sila ng first ever mammal from an adult cell, mammary gland. Yung pangalan niya, si Dolly the sheep. Narinig niyo na ba si Dolly? Familiar ba kayo kay Dolly? Hello? Hello? Familiar ba kayo kay Dolly?
Yes po, ma'am.
Okay,
Yes,
familiar na. Okay, mabuti naman at familiar kayo kay Dolly. So, titignan ko lang kung baka tulog na pala kayo. So, si Dolly, isa siyang sheep. So, yung ginawa kay Dolly, clone siya, di ba? From another adult sheep. And yung nucleus ng mammary gland doon sa pinagkuhanan, kinuha yung nucleus, tapos nilagay sa isang egg cell. Yung egg cell na yon from a surrogate mother na tinanggalan ng nucleus. So, ang gulo kong mag-explain. Wait lang. So, si Dolly, meron ka munang mammary gland galing sa pinagkuhanan nung clone, pinagkuhanan ng para i-clone si Dolly, yung original Dolly. Tapos, yung nucleus doon kinuha. Tapos, meron ka naman ngayon na egg cell. And yung egg cell na yon from a surrogate mother, yung egg cell na yon tinanggal yung nucleus doon sa egg cell na yon. So, nung tinanggal yung nucleus doon sa egg cell na yon, in-insert yung nucleus na galing doon sa mammary gland nung original Dolly. Tapos, yung yung egg cell na merong nucleus nung original na Dolly, nilagay doon sa isang surrogate mother. And nung pinanganak yung sheep, clone siya noong original sheep. And paano yun nangyari? Kasi yung kinuhang nucleus galing doon sa sheep na original Dolly. So, amazing actually tong event na to kasi nagbuka siya ng maraming possibilities and nabago niya yung ikot ng mundo. So, imagine niyo, di ba? Kunin niyo lang yung buccal cells na kunyari nang gusto niyong i-clone. Kunyari gusto niyong i-clone sino bang gusto mag-volunteer para maging clone? Volunteer ko na lang sarili ko. Kunyari gusto nyo i-clone ako. Tapos, kuha kayo ng kahit anong cell sa katawan ko. And yung pinakamadaling makuha ay yung buccal cell. So, kapag kinuha niyo yung buccal cell, kunin niyo lang yung nucleus. Tapos, kumuha kayo ng surrogate mother. Tapos, alisin niyo yung egg cell alisin niyo yung nucleus ng egg cell ng surrogate mother, tapos ilagay niyo yung nucleus ko. Pag pinanganak yung egg cell na yon na may nucleus ko, yung mapapanganak clone ko. Amazing, di ba? So, pwede kayo mag-clone. Pero hindi pa yun nagagawa, syempre. Meron yung ethical concerns. Nagagawa lang yung cloning ngayon sa animals. And itong bakit natin to kailangan i-mention? Parang nagbukas siya ng way para maintindihan natin yung eukaryotic gene regulation. So, kapag kaya mo mag-clone gamit ang kahit anong cell sa katawan, ibig sabihin, all cells of your body contains the same genomic material, right? So, lahat ng cells sa loob ng katawan mo, same lang yung genomic material. Pero, kung same lang yung genomic material sa lahat ng cells ng katawan mo, bakit may different types ka ng cells, di ba? Para siyang, basta. So, para masagot yon, titingnan natin yung yung gene regulation. So, kaya nag-give rise into different types of cells or different types of tissues dahil nare-regulate yung genes na na-express sa genomic material mo. Kahit pare-pareho lang yung genomic material sa loob ng lahat ng cells sa katawan mo. So, di ba? Hindi naman yung cells mo sa mata, hindi naman same yung genes na ina-express sa genes na ina-express sa cells mo sa lungs. And hindi rin yung genes na ina-express mo sa blood yung ina-express mo sa mata. So for example, sa RBC, di ba? Sa RBC, meron kang hemoglobin. And then na-express doon yung hemoglobin. Hindi mo naman kailangan i-express yung hemoglobin sa mata kasi hindi mo naman kailangan kumuha ng iron sa mata. Like, hindi mo naman siya ginagamit para mag-iron uptake, di ba? So, sana na-gets nyo. So, genomic control, ito again yung different ways to control in the level of the genome. So, i-mention ko lang ulit. Ayan. Pero, mag-focus lang tayo sa fun part. I-mention ko lang tong different types of genomic control mechanisms para hindi kayo mag-isip na ito lang na dalawa na i-discuss ko yung ginagamit for genomic control. So, yung i-discuss ko lang yung fun part, yung DNA methylation and histone modifications. And DNA methylation and histone modifications are very interesting. So, tingnan mo natin itong astronaut twins, si Scott and si Mark Kelly. So, si Scott and si Mark Kelly, twins sila na astronauts ng NASA. And since twins sila, you will assume that their genomic or their genome is almost the same or the same kasi nga twins sila. And merong experiment yung NASA, sinend nila si Scott Kelly sa space for 1 year. Tapos si Mark naman, nasa Earth lang for 1 year. And after 1 year, titingnan nila yung pagkakaiba ng genome ni Scott and ni Mark Kelly. And yung nakita nila after 1 year, maraming changes na nangyari. Isa sa naging changes kay Scott Kelly ay yung methylations ng DNA niya. And also, interestingly, mas humaba yung telomeres niya. So, ibig sabihin ba nun, mas mahaba yung buhay ni Scott Kelly kaysa kay Mark Kelly dahil humaba yung telomeres niya? Hindi natin alam. Sundan na lang natin sila. And ayun, so, pero bago ko i-discuss yung DNA methylation, ito muna, parang trivia lang. Si Scott Kelly kasi, sinusundan ko siya sa Twitter. Tapos, noong 2016, siya yung pinakaunang astronaut na nagpatubo ng flower sa space. Ito ay isang zinnia flower. Siya yung pinakaunang nagpatubo ng flower sa space. And interesting i-follow itong mga astronauts kasi kung ano-ano ginagawa nila. Like, minsan baliktad sila kumakain sa space. Tapos parang, basta kung ano, try nyo sila i-follow sa Twitter, ganun. Ayun, so i-discuss na natin yung DNA methylation and histone modifications. DNA methylation and histone modifications are part of this, under the so-called epigenetics. And what is epigenetics muna? Epigenetics is the alterations in gene expression that are transmitted from one cellular generation to the next without changing the DNA sequence. So epigenetics daw, you can alter how genes are expressed kahit na hindi mo binabago yung DNA sequence na yon, yung DNA sequence doon. Kahit di mo baguhin yung ATCG, blah, 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 pwede mo daw ma-alter yung gene expression. So, paano kaya yon? And also daw, itong mga epigenetic changes na to, it can be transmitted from one cellular generation to the next. Epigenetic changes involves DNA methylation and histone modifications. So DNA methylation, what it does is it actually represses or turns off your genes. So once your genes are methylated, they are turned off. Hindi mo sila ma-express. And ginagamit ito para kunyari, sa cells mo na hindi mo naman kailangan i-express yung certain kinds of genes, naka-methylate sila. And another type of epigenetic mechanism is in histone modification, wherein you modify your histones. Remember, your histones are responsible for packaging of your DNA, and you modify that packaging to open up your DNA to be able to express the DNA. And tingnan natin. DNA methylation is the addition of methyl groups to selected cytosine bases in the DNA. So mostly daw, yung methylation, nangyayari siya sa cytosine bases ng DNA. And again, yung function or yung parang pinaka nangyayari kapag mayroong DNA methylation ay na-turn off yung gene or nare-repress yung gene. So kapag may methylated yung genes mo, hindi mo siya ma-express. Another is histone modification. So first we have this so-called histone acetylation. In histone acetylation, what happens is that first meron ka dibang histones, tapos naka-wrap around doon yung DNA. Yun yung tinatawag mong nucleosome. Ngayon, kung kailangan mo mag-express ng gene na nandito, kailangan mo siya i-open up, di ba? So, paano mo i-open up itong packaging na to? Through histone acetylation. In histone acetylation, a histone acetyl transferase called HAT, an enzyme, it ina-acetylate niya yung histone. And kapag na-acetylate niya yung histone, it also allows transcriptional activators to bind to those areas or regions of the nucleosomes na merong acetylation. So therefore, acetylation, yung pinaka nangyayari ay turning on ng gene. So kapag nag-acetylate ka, matuturn on yung gene, ma-express yung gene. And another histone modification is through chromatin remodeling. In chromatin remodeling, you have a chromatin remodeling complex. Tapos, ina-expose niya basically yung DNA mo. And again, it allows the transcriptional activator to activate this region of the genome for the genes na nandito na magamit or ma-express. So, itong dalawang to ginagamit to turn on your genes. Merong video. Wait lang.
Here's a conundrum. Identical twins originate from the same DNA, so how can they turn out so different, even in traits that have a significant genetic component? For instance, why might one twin get heart disease at 55 while her sister runs marathons in perfect health? Nature versus nurture has a lot to do with it, but a deeper related answer can be found within something called epigenetics. That's the study of how DNA interacts with the multitude of smaller molecules found within cells, which can activate and deactivate genes. If you think of DNA as a recipe book, those molecules are largely what determine what gets cooked when. They aren't making any conscious choices themselves. Rather, their presence and concentration within cells makes the difference. So how does that work? Genes in DNA are expressed when they're read and transcribed into RNA, which is translated into proteins by structures called ribosomes. And proteins are much of what determines a cell's characteristics and function. Epigenetic changes can boost or interfere with the transcription of specific genes. The most common way interference happens is that DNA, or the proteins it's wrapped around, gets labeled with small chemical tags. The set of all of the chemical tags that are attached to the genome of a given cell is called the epigenome. Some of these, like a methyl group, inhibit gene expression by derailing the cellular transcription machinery or causing the DNA to coil more tightly, making it inaccessible. The gene is still there, but it's silent. Boosting transcription is essentially the opposite. Some chemical tags will unwind the DNA, making it easier to transcribe, which ramps up production of the associated protein. Epigenetic changes can survive cell division, which means that they could affect an organism for its entire life. Sometimes that's a good thing. Epigenetic changes are part of normal development. The cells in an embryo start with one master genome. As the cells divide, some genes are activated and others inhibited. Over time, through this epigenetic reprogramming, some cells develop into heart cells and others into liver cells. Each of the approximately 200 cell types in your body has essentially the same genome, but its own distinct epigenome. The epigenome also mediates a lifelong dialogue between genes and the environment. The chemical tags that turn genes on and off can be influenced by factors including diet, chemical exposure, and medication. The resulting epigenetic changes can eventually lead to disease if, for example, they turn off a gene that makes a tumor-suppressing protein. Environmentally induced epigenetic changes are part of the reason why genetically identical twins can grow up to have very different lives. As twins get older, their epigenomes diverge, affecting the way they age and their susceptibility to disease. Even social experiences can cause epigenetic changes. In one famous experiment, when mother rats weren't attentive enough to their pups, genes in the babies that help them manage stress were methylated and turned off. And it might not stop with that generation. Most epigenetic marks are erased when egg and sperm cells are formed. But now, researchers think that some of those imprints survive, passing those epigenetic traits on to the next generation. Your mother's or your father's experiences as a child or choices as adults could actually shape your own epigenome. But even though epigenetic changes are sticky, they're not necessarily permanent. A balanced lifestyle that includes a healthy diet, exercise, and avoiding exposure to contaminants may, in the long run, create a healthy epigenome. It's an exciting time to be studying this. Scientists are just beginning to understand how epigenetics could explain mechanisms of human development and aging, as well as the origins of cancer, heart disease, mental illness, addiction, and many other conditions. Meanwhile, new genome editing techniques are making it much easier to identify which epigenetic changes really matter for health and disease. Once we understand how our epigenome influences us, we might be able to influence it too.
Ayun. Ayun, so nakita niyo na yung, wait lang. Kita niyo na yung, ano sasabihin ko? Epigenetic changes. Epigenetic changes, interesting siyang aralin. Kasi at the level of the genome, kaya mong baguhin yung genomic, yung at the level of the genome, kaya mo siyang baguhin kahit na di mo babaguhin yung DNA sequences mo. So kahit na pwede mo gamitan yung DNA methylation and histone modification. So di ba outside yon ng genome niya. So di mo na kailangan baguhin yung sequences. So proceed na tayo sa level of transcription. So in the level of transcription, wait lang. So in the level of transcription or in transcriptional control, we have this so-called differential gene expression. So dito natin mas makikita kung bakit nagkakaiba-iba yung types ng cells na na-express Nawalan ako ng bumagal yung internet namin. Sorry. Wait lang. Recording pa naman. Mabuti di kayo umalis. Sorry. Sorry. Hello, naririnig niyo na ba ako?
Yes po, ma'am.
Yes po, ma'am.
Okay, thank you. Pasensya na bumagal yung internet namin. Yun. So, proceed na tayo. So, nasan na tayo? In the level of transcription, tayo, sa second level na. So, in the level of transcription, yung pinaka-distinguishing factor is the differential in the gene expression. And dito mo, mas makikita kung paano nagkakaroon ng iba-ibang cell types. Kasi, iba-iba yung kung paano nagtatranscript yung mga cells, kung ano yung pinuproduce nilang mRNA. And in the level of transcription, yung pinaka-nag-control ay yung transcription factors. And kung naalala nyo, yung video doon sa DNA replication, di ba, nung pinapanood ko sa inyong video, meron pa folding na nangyari. Parang nag-fold yung DNA. Tapos bakit kaya biglang may folding na nangyari bago mag-start yung transcription? Sana naalala niyo yung video. Kung hindi niyo maalala, panoorin niyo ulit. Mayroong pa folding na nangyari na naganap sa unahan. So yung nangyari doon, actually, yung enhancer region, yun yung parang nagko-control nung kung kailan magkakaroon ng transcription doon sa promoter region. So what happens is that yung merong activators na tinatawag, and the activator proteins, they bind to the enhancer, triggering DNA bending. And DNA bending, what happens here is that the activators, they interact with co-activators to stimulate chromatin remodeling and histone acetylation. So, ito, remember, ay epigenetic factors, chromatin remodeling and histone acetylation, to expose your DNA. So, kapag na-expose mo na yung DNA mo, yung activators, they bind to mediator triggering assembly of RNA polymerase and general transcription factors at the promoter site. So, sabi daw dito, kapag na-expose na yung DNA mo, and meron ka ng activators, magkakaroon ka ngayon ng mediator, and yung mediator na yun, magti-trigger na siya ng pag-bind ng RNA polymerase and ng mga transcription factors doon sa promoter region nung ito-transcribe mong gene. So, ayun yung transcriptional control. Involved doon, again, yung enhancers mo and yung activators mo. So, kapag pinanood nyo ulit yung video ng transcription nung una, merong folding na nangyari sa unahan. Balikan nyo yan. And ngayon, meron tayong mga techniques to see how RNA is transcribed or kung ano yung RNA na express ng mga cells. So, the first technique is northern blotting. Narinig niyo na ba yung northern blotting? Hello? Narinig niyo na ba yung northern blotting or di pa?
Di pa po.
Okay, di pa raw. So, yung northern blotting, common siyang technique in molecular biology to see the expression of RNA. So, meron tinatawag ka rin na southern blotting. Yung southern blotting naman, siya naman yung sa DNA. Yung western blotting rin, meron ka rin tinatawag na western blotting. Western blotting sa, hulaan nyo, sa proteins. So, ang galing nung nagpangalan nitong mga blotting techniques. Northern blotting is for RNA. Southern blotting is for DNA. Western blotting is for protein. Pero northern blotting yung titingnan natin ngayon. And in northern blotting, first you have your sample. Titingnan mo kung ano yung RNA na ina-express doon sa sample. So you extract the RNA and then you subject it into electrophoresis. And after subjecting it into electrophoresis, itong electrophoresis, ituturo ko to sa lab nyo, huwag kayong mag-alala. Electrophoresis, tapos ito transfer mo siya into membrane or dito mo na gagawin yung northern blotting. So kapag transfer mo siya doon sa membrane, ililabel mo na siya ng probes. And you will visualize now yung RNA mo sa X-ray film. And under the X-ray film, you will see which RNAs are expressed. Makikita mo yung probes mo kung ano yung bands na gagawin nila. And based doon sa bands na makikita mo doon sa X-ray film mo, doon mo makikita kung ano yung RNA mo na in-express. And ito yung medyo manual na pagtingin kung ano yung in-express ng RNA. Pero merong mas maganda, mas advanced. Tinatawag natin microarrays. Microarrays, they show how genes are expressed differently in various cell types. And they can be used to investigate cancer cells for targeted therapy or personalized medicine. So, isa-isahin natin. So, in microarrays, say for example, you want to see which genes are expressed in your brain cells and in your liver cells. Trip mo lang i-compare yung brain cells at liver cells. So, yung una mong gagawin, i-isolate mo yung mRNA nila. And after mo ma-isolate yung mRNA nila, gagawa ka ng cDNAs or complementary DNAs via reverse transcriptase. And i-label mo sila ng fluorescent dyes. And for brain cells, yung naisip mong label ay green, kulay green. And sa liver cells naman, yung nilabel mong fluorescent dye ay red. Tapos ngayon, makatapos mo sila i-label ng fluorescent dyes, ilalagay mo na sila sa isang tray called your microarrays. Tapos, meron dun na wells. So, isipin nyo, itong microarray, para siyang yung ice tray sa ref, di ba? Yung parang sa ice cube, lalagyan ng ice cube. Kada well doon, sa tray na yon, merong specific gene. So, for example, yung isang well, specific gene siya for the production of hemoglobin. So, kung nilagay mo yung sa well na yon, sa well ng may gene for hemoglobin, yung brain cell and liver cell, walang expression. Wala kang makikitang kulay nung fluorescent dyes kasi hindi naman nag-express ng hemoglobin yung brain cells and liver cells mo. But, if you put your brain cells and liver cells in a gene na common sa kanilang dalawa, for example, albumin, gene for albumin, pag nilagay mo yung both brain cells and liver cells doon, yung magiging kulay ay kulay yellow. Ito. So, ibig sabihin, pag yellow, parehong cells nag-express ng gene na yon. And if green lang yung kulay, ibig sabihin, brain cell lang yung nag-express ng gene na yon. Pero kapag red lang yung kulay, ibig sabihin, liver cells lang yung nag-express na yon doon. So, meron ka ngayong 4 na kulay na titingnan. Number 1, green. Kapag green, brain cells lang yung nag-express ng gene na nandun sa well. Kapag red, liver cells lang. Kapag yellow, both cells express those genes. For example, yung albumin mo, parehong nag-express yung brain cell and liver cell mo ng albumin. And lastly, kulay black. Ibig sabihin, kung black lang, wala siyang ina-express. Yung gene na yun, hindi siya ina-express ng both brain cells and liver cells. Okay, and this microarray, actually, you can use this to investigate cancer cells for targeted therapy or personalized medicine. So, kung magpupunta na kayo ng med school or maging kayong doktor, pwede ito gamitin para i-treat yung cancer patients nyo. Bakit? Kasi, makikita mo, kapag kinuha mo yung cancer cells from your cancer patients, tapos ilagay mo sila sa microarray, makikita mo kung ano yung genes na expressed at hindi na expressed. So ngayon, alam mo kung ano yung genes na nandoon sa cancer cells mo. Therefore, mas malalaman mo kung alin yung dapat mong i-target and ano yung pwede mong gamiting gamot doon sa cancer cells na yon or sa cancer patient. So maganda siyang gamitin. Next level naman natin ay in the level of the post-transcriptional control na tayo sa RNA processing and nuclear transport. And also translation. And lastly, post-translation. And tingnan natin sa post-transcriptional control sa level po ng RNA processing and nuclear transport. remember niyo yung alternative splicing, di ba? In-explain natin yung alternative splicing doon sa transcription and translation. And yung alternative splicing, isa siyang form ng regulation of genes mo rin. Kasi you can splice your RNA and form different kinds of different types of the protein. So, example natin dito ay yung IgM. So, IgM, again, yung alam natin sa IgM, antibodies siya kapag may bagong infection. Para maalala niyo yung IgM, trick lang. IgM, sunod na letter N, ibig sabihin now. IgM, now, ngayon yung infection. IgG, sunod na letter H, history. IgG, yung antibodies na yon ginagamit kapag matagal na yung infection. So, trick lang. And ito, dito balik tayo sa alternative splicing. So here, as you can see, itong first na alternative splicing, merong transmembrane anchor. And different yung pagka-splice ng pre-mRNA niya. So therefore, yung na-produce niyang IgM, meron na transmembrane anchor and naka-bound siya doon sa membrane. And dito naman sa isang splicing variety, walang transmembrane anchor. Tinanggal yung transmembrane anchor dito. Therefore, yung IgM dito ay secreted or free sa cells mo. So, ayun lang yung alternative splicing, part siya ng post-transcriptional control. And the level of the translation, what you see here is kung paano mo i-control yung translation. So, yung example dito ay yung paano kinokontrol ng iron, yung RIE binding proteins or iron response element proteins. So, dito sa first, mababa yung iron concentration. Therefore, yung IRE or iron response element mo naka-hairpin loop. And kapag naka-hairpin loop siya, hindi niya inaalaw yung binding protein para mag-pass through para ma-express itong ferritin mRNA. Okay. Pero kapag merong iron doon sa loob ng cell, magbabind ngayon yung iron doon sa binding protein. And ma-allow na ngayon yung ribosome to translate yung mRNA mo. So, ayun. Sa level yon ng translation. And ito, post-transcriptional control naman, ito yung favorite part ko actually, sa RNA interference or RNAi. Dito tayo mag-focus. RNAi or RNA interference from the word itself is based on the ability of small RNAs to trigger mRNA degradation or inhibit mRNA translation or inhibit transcription of the gene coding for a particular mRNA. Ang haba, di ba? Pero hindi naman yan kailangan i-memorize. Yung kailangan nyo lang tandaan ay RNA interference ay para mag-interfere ng RNA, di ba? Ang galing. Yun yung nasa pangalan niya na. RNA interference para mag-interfere ng RNA. Specifically, messenger RNA. So, ano ba ito nangyayari? So, we have first yung small interfering RNA mo called your siRNA. And yung siRNA, yung interesting dito is you can actually design your own small interfering RNA. You can design your own double-stranded RNA, insert it into your cells, and then you can inhibit the translation of a certain mRNA. So, pwede kang mag-target ng mRNA, pwede kang mag-design ng double-stranded RNA, and i-target mo yung specific mRNA. So what happens first is, once you insert a double-stranded RNA, a protein called dicer cleaves the double-stranded RNA into a small interfering RNA about 21 to 22 base pairs in length. So yung dicer, madali lang siya tandaan kasi dinadice niya yung double-stranded RNA tapos pinuputol niya. Tapos gumagawa ka na ngayon ng small interfering RNA. And this siRNA or small interfering RNA combines with the RISC proteins. And yung RISC proteins mo, complex siya ng proteins, including a slicer, a thing called slicer. And once mag-bind na yung siRNA mo sa RISC proteins mo, makakabuka na ngayon nitong buong complex na to called your C-RISC, small interfering RISC. Ito. And matatanggal itong nasa bottom part. Tatanggalin itong sa bottom part. So therefore, magkakaroon ka na lang nito ng parang comb. Parang comb siya ng RNA. Tapos meron kang nakabound na proteins. And makikita nyo na kapag itong RNA na to, itong interfering RNA na to, nahanap niya yung mRNA na specific na gusto mong i-interfere, gusto mong i-stop yung translation. Either two yung magiging action niya. So, either of the two yung magiging action niya. If exactly na complementary yung siRNA mo doon sa mRNA mo, na target mRNA mo, madedegrade yung mRNA or masisira yung mRNA. Therefore, kung wala ka ng mRNA, wala ka ng translation into proteins, di ba? Di na ma-express yung proteins. Pero kung partly complementary lang yung itong complex mo dito sa target mRNA mo, magkakaroon ka lang ng inhibition. Hindi masisira yung mRNA mo. Okay. So, yun yung small interfering RNA. And isa siyang way sa RNA interference. And itong small interfering RNA, pwede siyang either naturally occurring in your cells or pwede mo rin siya i-design. So, amazing kasi pwede ka mag-design ng RNA na pang-target ng mRNA sa katawan mo para hindi mag-produce ng protein yung cells na target mo, kung ano man yung trip mong i-target. Next naman ay naturally occurring RNA interference in our body. Naturally occurring to kasi merong genes sa DNA mo mismo siya, pero pinaproduce na miRNA. Ito yung tinatawag nating microRNA or miRNA. So first, nagpo-produce ng pre-miRNA or primary microRNA. Tapos, meron kang protein na draw siya, tinatawag na draw siya, and i-cleave niya yon. Para lang rin yung dicer kanina, di ba? Kinat niya yung RNA, yung double-stranded RNA. Tapos, after makat, lalabas na yung pre-mRNA, and dito na pala yung dicer. Magkakaroon ulit ng cleavage, aayusin mo kung alin lang doon sa pre-mRNA yung gagamitin mo. So, para lang rin siyang splicing. Parang splicing lang. And after ma-export na siya sa cytoplasm, yung mRNA mo, yung microRNA mo, again, it will complex with your risk proteins, itong proteins na to, which contains a slicer. And if partly complementary lang, again, yung messenger RNA mo, inhibition lang yung mangyayari. Pero kung exactly complementary or pareho yung sequences nung ginamit mo, nung nagamit na microRNA doon sa target messenger RNA, yung slicer protein, i-degrade niya yung messenger RNA mo. So, halos same lang yung principle doon kanina sa small interfering, di ba? Kapag partially lang na complementary, inhibition lang. Pero kapag perfectly or exactly complementary yung microRNA sa messenger RNA mo, i-degrade niya yung mRNA mo. Therefore, wala ka ng translation ng protein. And ayun. So, merong video. Last video na to. Malapit na matapos. Ayan yun. Wait lang. So, kung may tanong kayo, guys, magtanong lang kayo.
You can think of your cells as the kitchen in a busy restaurant. Sometimes your body orders chicken. Other times it orders steak. Your cells have to be able to crank out whatever the body needs, and quickly. When an order comes in, the chef looks to the cookbook, your DNA, for the recipe. She then transcribes that message onto a piece of paper called RNA and brings it back to her countertop, the ribosome. There, she can translate the recipe into a meal, or for your cells, a protein, by following the directions that she's copied down. But RNA does more for the cell than just act as a messenger between a cook and her cookbook. It can move in reverse and create DNA, it can direct amino acids to their targets, or it can take part in RNA interference, or RNAi. But wait, why would RNA want to interfere with itself? Well, sometimes a cell doesn't want to turn all of the messenger RNA it creates into protein, or it may need to destroy RNA injected into the cell by an attacking virus. Say, for example, in our cellular kitchen, that someone wanted to cancel their order or decided they wanted chips instead of fries. That's where RNAi comes in. Thankfully, your cells have the perfect knives for just this kind of job. When the cell finds or produces long, double-stranded RNA molecules, it chops these molecules up with a protein aptly named dicer. Now, these short snippets of RNA are floating around in the cell, and they're picked up by something called RISC, the RNA silencing complex. It's composed of a few different proteins, the most important being slicer. This is another aptly named protein, and we'll get to why in just a second. RISC strips these small chunks of double-stranded RNA in half, using the single strand to target matching mRNA, looking for pieces that fit together like two halves of a sandwich. When it finds the matching piece of mRNA, RISC's slicer protein slices it up. This cell then realizes there are odd, strangely sized pieces of RNA floating around and destroys them, preventing the mRNA from being turned into protein. So you have double-stranded RNA, you dice it up, it targets mRNA, and then that gets sliced up too. Voila! You've prevented expression and saved yourself some unhappy diners. So how did anybody ever figure this out? Well, the process was first discovered in petunias, when botanists trying to create deep purple blooms introduced a pigment-producing gene into the flowers. But instead of darker flowers, they found flowers with white patches and no pigment at all. Instead of using the RNA produced by the new gene to create more pigment, the flowers were actually using it to knock down the pigment-producing pathway, destroying RNA from the plant's original genes with RNAi and leaving them with pigment-free white flowers. Scientists saw a similar phenomena in tiny worms called C. elegans, and once they figured out what was happening, they realized they could use RNAi to their advantage. Want to see what happens when a certain gene is knocked out of a worm or a fly? Introduce an RNAi construct for that gene and bam, no more protein expression. You can even get creative and target that effect to certain systems, knocking down genes in just the brain or just the liver or just the heart. Figuring out what happens when you knock down a gene in a certain system can be an important step in figuring out what that gene does. But RNAi isn't just for understanding how things happen. It could also be a powerful therapeutic tool and could be a way for us to manipulate what is happening within our own cells. Researchers have been experimenting with using it to their advantage in medicine, including targeting RNA in tumor cells in the hopes of turning off cancer-causing genes. In theory, our cellular kitchens could serve up an order of cells, hold the cancer.
Yun. Okay. So, ayun yung RNAi. Wait lang. Meron ba kayong tanong so far tungkol sa RNAi? Interesting yung RNAi actually. Kasi nga, pwede ka mag-design ng RNA. Tapos, target ka nung unwanted mRNA na gusto mong, na ayaw mo i-express, di ba? So, pwede mo yung gawin research. So, ayun. So, last na, for our F protein in HIV. So, ito yung parang kinuha kong parang case study or parang para ma-relate nyo yung control of gene expression into a disease. So, there's this thing called Rev protein in HIV, which functions as the regulator of expression of virion protein, and it mediates the cytoplasmic transport of viral mRNA. So, medyo intimidating tong figure, di ba? Huwag niyo pansinin tong nasa baba. Ito lang yung pansinin niyo, tong Rev. Tong Rev protein na to, important siya. Bakit? Kasi in patients with HIV, bago ma-express yung HIV ng nucleus, kailangan muna nung mRNA na ma-transport from your nucleus to your cytoplasm, di ba? Para ma-express siya. And amazingly, merong protein na involved sa transfer ng mRNA ng HIV from the nucleus to the cytoplasm. And yung tawag doon ay Rev protein. So if you control the Rev protein, you also control the expression of the HIV. So if you interfere with the Rev protein from transferring the mRNA of the HIV, of the virus from the nucleus to the cytoplasm, hindi ka magkakaroon ng translation, and in turn, hindi ka rin magkakaroon ng viral particle assembly. And way rin to para ma-lower yung viral count sa patients with HIV. So, ayun lang. Yun yung isa sa mga application. And sobrang daming application ng control of gene expressions. Pwede nyo silang magamit kung ano mang trip yung disease, yung gusto nyong gamutin. So, ayun. To summarize, we learned about the different control of gene expression in bacteria and eukaryotes. We also investigated some techniques to measure gene expression, such as northern blot and microarrays. And we also explored how the knowledge on the control of gene expression mechanisms can be applied in treating diseases. Example, yung HIV and also yung cancer. Pwede mong gamitin yung learnings nyo dito para mag-treat ng cancer patients balang araw. So, yun lang. Yung control of gene expression, meron ba kayong patanungan? Meron ba kayong tanong? Meron ba kayong tanong?